General Biology, Chapter 1, Part 2. The importance of ethics in science can't be overstated. In fact, we often have to be reminded how important it is to be honest and to perform all of our work every day with integrity. What does that mean? Integrity essentially means not falsifying or using non-valid concepts. By valid, we mean that we can use particular steps to evaluate and reveal what is or isn't the truth, or what is or isn't real. How do we know that? Well, we're somewhat limited because we can only use our own physical senses or biological senses to truly determine what is or isn't real. And that is our limitation. But that's what we have to work with, and as long as we're revealing and using our best possible interpretation or revealing or documenting what we've observed with our senses, then that's the best we can do. For scientific ethics, this is applied to all stages and all disciplines of science, both the biological and physical sciences. We observe and record what we see, feel, hear, touch, use all of our senses to evaluate. We record that information in the form of what we call data or data. And the data are numbers, measurements, observations, and they're as accurate as we could possibly make them based on the technology that's available to us at the time. We need to share that information or those findings properly and share them with our colleagues or folks who are also interested in the same concepts or observations, and then include those observations and findings in what we would consider unbiased scientific resources, books, journals, the whole broad pool of scientific knowledge. Publications of data, again, measurements, observations, all kinds of results from research projects are generally published in what we call peer-reviewed or colleague-reviewed, folks who are going to have the same expertise that we do that are performing the research or the investigation, in books and what we call journals, which are scientific magazines. And those particular studies, whether they're just experimental projects or observations in nature, someone needs to go out and replicate and find the same results that we did. That is the problem today. There's not nearly enough peer-reviewed replication of studies that have been done. There's so much emphasis and so much pressure on scientists today to do essentially what would be described as primary studies, where they're the first to find or the first to make those observations, that there's less and less replication or redoing of those same experiments, or going out into the forests or the jungles or the deserts to make those same observations and come up to the same conclusions. That sometimes we're not getting the type of integrity that we need in good scientific sources of knowledge. The only thing we can do is encourage everyone that can to go out and double check, make sure they're replicating or repeating those same procedures and see what they come up with. Collaboration is really required by scientific ethics. We need to work well with others. Regardless of what our other kinds of concepts or other sort of belief systems might be, science is not a religion or a faith-based assembly of knowledge. Scientists really do try to be objective, or at least they should. Objective means we're not including our own thought bias or our own biases in our observations. We're just reporting the facts, just the facts of what we see, feel, touch, smell, measure. Again, using our senses as unbiased or ethically as possible to find some truly valid observations, raise questions, answer questions, and continue to investigate this beautiful world around us. There's a philosophical branch of study called logic. Logic really is a good foundation to use as a scientist. It is the foundation for what's described as scientific reasoning. How do we determine what is or isn't around us? And there are two general categories of logic: inductive reasoning and deductive reasoning. With inductive reasoning, it's a form of logical thinking that uses related observations to arrive at a general conclusion. Once you've established that general conclusion from all of those numerous observations, then you can actually use that general conclusion to establish some deductive reasoning and make some predictions about what you think might also happen with some other entities or other scenarios. So the general conclusion from inductive reasoning, multiple observations coming to a general conclusion, can then be used for the next branch of logic, which is deductive reasoning. You use that general conclusion to forecast or predict what should happen based on that general principle. We now call these general biology theories, or sometimes they're called natural laws, which seems to be how nature and how our world works, based on thousands of years of observations and thousands of even modern observations and documentation of how the world seems to really work. So the two types of reasoning go hand in hand. With inductive reasoning, again, we have a number of observations or tests that are performed in the laboratory, and for example, members of a species are not all the same. Individuals compete for resources. Species are generally adapted to their environment, and then we can come to some kind of general conclusion. That general conclusion then can be the general premise for further deductive reasoning to predict something about the particular environment, climate, individuals who are living there, and the two types of reasoning go hand in hand. The idea is to use a very sort of precise, unbiased, step-by-step evaluation for the inductive reasoning component, and then again use the same kind of unbiased, predictive nature of the deductive reasoning to try and determine what's around us and how our world and the organisms on it organize and work together. For thousands of years, our ancestors have worked to try to establish a step-by-step process to, again, using logic, evaluate their environment. It wasn't until about the middle of the 1600s, during the 17th century, that a scientist by the name of Francis Bacon developed a step-by-step process for inductive reasoning that could be used in the sciences to evaluate what's around us. It consisted of several well-defined steps and is observed or recognized today as scientific method. Now, the scientific method is a very valid, well-established, well-tested process, and is a very formal process. And most scientific investigation today doesn't often start out using these very formal steps of the scientific method. But when we have a question, so we've made an observation in nature, and we have an observation that develops or leads to a question in our mind about, or even in our cities, towns, groups, just some kind of question about nature perhaps. And we try to figure out, well, how can we answer that question? Well, we can develop essentially an informal, testable question, a hypothesis. Now, a hypothesis in science is a term that's used a little bit differently than, let's say, if you're reading a novel or perhaps even looking at a crime scene and you hear someone say, I have a hypothesis about how this might have happened. A hypothesis really is a term that means a testable question, and in biological sciences, that means the test is some type of experiment or procedure that can search for the answer to our question. Oftentimes, an informal hypothesis is established. We ask that question, and based on that question and the types of experiments or procedures we set up to try and answer that question, we may have to modify or adjust that initial question or hypothesis so we can zero in or fine-tune our experimental design to more appropriately reveal the results or answer to our question. Eventually, we can establish a very formal hypothesis that's the root of the scientific method. It is the foundation of a good scientific method, where we have a good hypothesis that's leading us to a supported or non-supported result or hypothesis. If it's a supported hypothesis, and if we can repeat this procedure over and over and over thousands, tens of thousands, or maybe even millions of times, eventually a well-supported hypothesis to an observation made on Earth turns into, at least for the biological sciences, a theory. So a theory is a well-supported, well-documented hypothesis that has a lot of data to say, yes, this is how things seem to work, or this is how we observe the natural world and what's going on. If the hypothesis is not supported, or essentially we get a no at the bottom of our series of procedures or investigations, then we have to go back and reevaluate, how do we restate this, or what went wrong? And how do we keep digging or performing different procedures and experiments to eventually get to what we would define in general, what is real, or what is the truth? What are the sources of errors that might have occurred? Or what are the sources of errors that still might occur, even if we have a well-supported hypothesis or theory? What are the challenges? Has there been any what we call quality control? Through every step of any procedure or research experiment or process, there should be a constant evaluation of the integrity of the procedure, the integrity of the materials used. Were the materials fresh? Were they properly maintained? Was the equipment or the technology that was used the appropriate type of technology? All of these concepts must feed into the actual real process of a scientific investigation. So even though scientific method on paper looks like a fairly simple, straightforward process, and in sort of these general concept it is, the actual, where the rubber hits the road, when you actually do these steps, takes a lot of time, effort, careful thought, and it must be done with the highest level of ethics and integrity to make certain that we are trying to reveal what's really around us and how organisms interact with each other and the world. Just like we have two general types of science, the biological and physical sciences, in the areas of sciences in general, we also have two types or groups that can be divided into basic versus applied. Basic science or pure science just simply tries to add to our knowledge base of any particular discipline. It's just investigating what's there and what isn't there, what works and what doesn't work. Applied sciences often use information from the basic science to solve real-world problems. And they overlap again very nicely. Basic science or pure science often as simply is investigating just some general concepts or maybe some very specific concepts with no particular goal in mind except to answer the question of what is this, or how does it work? What's the structure? Where does it live? What does it eat? The applied sciences then can often go back into the literature and start to apply or take advantage of the results and find a useful application. There's nothing wrong with science being useful. And in fact, the scientific publications that are available for us to read and gather up new knowledge and take that new knowledge, especially from the basic sciences, and perhaps transfer that information to the applied sciences, often requires publications in the form of journal articles or textbooks or even regular just reading books in the peer-reviewed process, which means our colleagues read and evaluate what we've done and give us some feedback. Many of the magazines, or what we call scientific journals more specifically, follow a format called the IMRAD format. IMRAD is an acronym that stands for the sections or the parts of a scientific paper or a journal article. It starts out with a synthesis of the whole entire paper or the whole entire report called an abstract. The content of the abstract actually is the most difficult portion of a scientific paper for students to formulate themselves. I call it a Reader's Digest version of the journal article. If you don't know what a Reader's Digest is, many students don't anymore, it's again essentially a condensation version of the whole paper, taking a single sentence to distill or concentrate the whole rest of the results. It also includes an introduction that gives folks a little background, the materials and methods that were used to perform the process or project, the results or the findings of the experiment, a discussion of interpreting the results, and then perhaps a conclusion and the literature cited. More in-depth descriptions of how we perform or organize our papers using this IMRAD format. So there's a lot more detail to it, and it is what we would expect in a publication for a scientific journal article. A good, easy, recognized application of the basic sciences to applied sciences can be seen when, let's say, we have some kind of disaster on Earth and wildlife is affected. Natural scientists or biologists who go out in the field and simply study different animals and plants just to learn about how they interact with each other and with the rest of the world has been used as a source of basic science, knowing how different little organisms or big organisms live. Then we could take that knowledge, and it actually has been done, and apply it whenever some type of disaster occurs to return these organisms to their healthy environments. So animals that were affected by hurricanes that destroyed their habitats, or unfortunate oil spills that destroyed their habitats, then scientists can use the knowledge of how their habitats or where they lived should look and how clean these organisms should be, or where they build their nests or what types of foods they eat. All of that basic science information can be used to what we call rehabilitate or return the animals and plants to their health and healthy environments. In the biological sciences, we have different properties that we assign to organisms or things and entities that we see here on Earth and call organisms. There is a hierarchy of organization or sort of characteristics that we would assign to an organism to say this is a biological entity. And the three main criteria that we're going to use as biologists to recognize something as an organism, whether it's here on Earth or any other body or planet or moon. By body, I mean some kind of structure in the solar system or the galaxy. And those three criteria that we're going to recognize and identify something as an organism is essentially adaptation, responsiveness to stimuli in its environment, reproduction, making copies of itself or having offspring, and growth and development. In other words, some form of metabolism where they're consuming some kind of nutrient and growing, and then, of course, using that growth and development for all those other two criteria, reproduction and adaptation. Three additional criteria that are often recognized in the biological sciences to identify an entity as an organism is regulation, in other words, controlling or interacting with the environment and maintaining what we call homeostasis, or a balance of their growth and reproduction and their metabolism, and some energy processing, which again is part of what's happening with growth and reproduction and adaptation, using some sort of what we call energy to do things. The hierarchy of organization of organisms starts with the tiniest, not easily split or divided structure that we call an atom. Atoms are organized into larger structures called molecules. Molecules are organized into larger structures called macromolecules. Those macromolecules are used to create organelles. Organelles are some of the smaller structures that are associated or assembled together in what we call cells. Cells, when they're organized together, are creating tissues. Tissues can be found in various structures in some of the organisms that we call plants and animals, that we see as organs and organ systems. Those organ systems are found in organisms that live together in populations, and then communities, ecosystems, and then eventually what we call the biosphere. And those are some of the concepts that we'll talk about off and on all semester.